All right. Welcome, everyone. And thank you for joining us for the first session in our BmDR workshop series. Hopefully everyone can hear me now. I know we usually give everyone a few minutes to join, but since we have so much information today, we're going to go ahead and get started right away. So my name is Katrina Perrone, and I'm an associate director at the Critical Path Institute within the Predictive Safety Testing Consortium, PSTC, and that's where the Biomarker Data Repository is housed. We're delighted to have you with us today as we introduce this resource that's designed to drive improvements in kidney health. This workshop launches a twice-yearly series, and we look forward to advancing this important work together. Before we begin, a few quick housekeeping notes. Participant lines will stay muted, so please use the chat for comments and submit questions for our panelists through the Q&A function in your Zoom toolbar. They may respond in writing in the Q&A or during the live discussion. And finally, today's session is being recorded and will be shared afterwards. Here's an acknowledgement that this work was supported in part by the FDA and non-government sources. Content reflects those of the authors and speakers, but not necessarily those of the FDA or the US government. And we'll begin with a brief BmDR 101 overview, followed by Alexander Deagle sharing his experience with chronic kidney disease and the importance of patient empowerment. Then Dr. Joseph Bonventry will discuss how BmDR is advancing the field. Andrew Palillo will review available data sets and biomarkers. And Dr. Patrick Murray will demonstrate how these data sets can be applied and the value of a centralized repository. We'll wrap up with a live panel discussion moderated by Dr. Gary Friedman. And with that, we'll go ahead and get started. I will kick us off with BmDR 101. So, what is the Biomarker Data Repository and why are we all here today? The BmDR is a publicly accessible resource that compiles safety biomarker data from multiple sources to advance the real-world understanding and use of emerging drug-induced kidney injury biomarkers, so DICKI. By enabling analysis across diverse populations and across kidney disease stages, it allows qualified researchers to explore new applications in clinical practice and drug development. Ultimately, this resource can help support future regulatory qualification efforts and strengthen the use of biomarkers to improve kidney safety assessment. The need for BmDR grew out of several urgent gaps in kidney health. We still lack reliable tools to detect kidney injury. Current clinical measures haven't changed much in decades. This makes clinical trials longer, more costly, and more difficult to interpret, especially when we can't easily determine whether a therapy is helping patients or causing harm. And because acute kidney injury is common and associated with serious long-term consequences, there's a clear need to accelerate the development of new treatments. BmDR can help with this by acquiring more data to help make faster, better informed decisions. It enhances collaboration across research groups, and most importantly, it integrates the patient voice to ensure tools focus on outcomes that matter, like delaying dialysis or transplantation and preserving quality of life. A quick review of what a kidney biomarker is. Kidney biomarkers are signals in your blood or urine that tell us how well the kidneys are working. They help detect problems earlier, monitor disease, and guide treatment. Tracking these biomarkers also helps patients better understand their health and participate in decisions with their care teams. BmDR is initially focused on the kidneys by bringing together data sets that include these biomarkers across different ages, genders, ethnicities, and types of kidney disease. This gives researchers a clearer picture of how biomarkers behave in diverse populations and creates a strong foundation to expand into other diseases in the future. In 2018, CPATH qualified a composite measure of kidney safety biomarkers that can be used alongside standard tests to help detect renal tubular injury in phase 1 healthy volunteer studies when a drug has a known risk. That context of use, or COU, focuses on population level safety monitoring. We are now pursuing a second COU that uses a biomarker panel to identify DICKI in individual patients, again, alongside standard tests. This expands the applicability beyond that cohort level assessment. By aggregating larger and more diverse data sets, the BmDR can strengthen the evidence for these two COUs. It can support future expansions and potentially inform new COUs, including those related to efficacy or recovery from kidney injury. Overall, the goal of the BmDR is to accelerate safer drug development by creating a unified ecosystem for biomarker knowledge. And by centralizing a high-quality biomarker data and making it easier for researchers to assess and analyze, BmDR helps improve the way treatments are evaluated, ultimately enabling safer, more effective therapies to reach patients faster. And what makes BmDR especially powerful is that it's not just a data storage center. It helps researchers compare across studies, populations, and biomarker types in a way that wasn't possible before. And this ability to see broader patterns and connect previously siloed information strengthens both the scientific understanding and real-world impact. Here's a quick look at the structure of BmDR and its committees that are made up of different community members. I won't detail them here, but if you have questions or are interested in joining any of these committees or contributing data to the BmDR, we'd love to connect. My contact information is shown here on this slide. And before we hear from the rest of our speakers today, here's a reminder to submit questions in the Q&A function so that we can get them answered. And that's all I've got for an intro. Thank you, everyone. And I will welcome Alexander Deagle, CPATH's Communication Director, to discuss his experience living with CKD. And let me give him, okay, Alex, you should be able to go.
Sorry, I'm getting the can't share video now because
No,
it is
because I
disabled by
want
the
to.
host. Yep, thank
Okay.
you. Just got it. All right. Starting here. Oh, you've asked me. Okay, start. Oh, there I am. Sorry about that. All right. Thank you, Katrina, for that warm welcome and introduction. I am Alexander Deagle, or Alex Deagle, with the Critical Path Institute CPATH team, Assistant Director of Communications here. Mostly on my day-to-day, I work and manage our website, write our blogs, our impact stories. I did want to get more into public speaking at the next phase of my career. I didn't think I would have to get a chronic kidney disease to do it, but here I am. I'm happy to be here for what it is, and happy to talk with you all about living with CKD and the value of being an empowered patient. So, quick background, my diagnostic odyssey, 37-year-old father of two, still an active athlete. I currently play for my hometown team of Old Gaelic Rugby in Mechanicsburg, Pennsylvania. For those of us in the DC area, I spent 6 years living in DC playing for a D1 team down there. Highlight of my career was I was captain of a team that drew from the top players from Raleigh, North Carolina to Pittsburgh, Pennsylvania. It was a traveling all-star team. Played in some professional matches, played against countless internationals, Olympians, including a two-time World Player of the Year, Perry Baker, who I'm happy to report only scored on us one time. So again, active, healthy, every physical, doctors clear me, say you're a healthy guy. Um, this past February, I was feeling very sick after day 5 of the fever that I had what ended up being influenza type A back in February. I was very much being a toxic male about the situation, messing with Mike Minchek, who's our PMO director. I can work through anything, 100-degree fever, no big deal. Day 5, 102. He's like, dude, go to the hospital. So I did, and it was a happy accident, or just a happy discovery. A urinalysis was conducted as part of my intake. They were quickly came to me and said they're seeing concerning amount of blood and proteins were found in the urine. As I said up front, I am not one of the doctors here at CPATH, but I knew enough to say that doesn't sound good. Scans and tests quickly ruled out the scariest options, so there's a little bit of a quick relief there in terms of liver or kidney damage, cancers, but still no diagnosis, right? You're telling me everything's functioning, and yet my filters are leaking. So what's going on here? So I had outpatient follow-up, another urinalysis. They're seeing the same thing, so that eliminated what they were hoping for, was just that I was so sick that, my body was just jacked up and work acting weird. So I was like, okay, well, you're seeing what you saw in the hospital. Nothing's getting better. Like, what is the plan here? Oh, well, you're so healthy otherwise, and you're young, you're strong. We're gonna wait and see. So, in the springtime is when we ramp up for playoffs, and we were fortunate enough to go to the championship game that year. So, I'm still trying to live my life, still trying to be an athlete, still trying to be a rugby player, which has been a part of my life since I was 17 years old and first picked up a rugby ball. We have this championship game in late April. I'm playing the playoffs through the championship. I'm wondering, like, should I even still be out here? Like, I know something's not right inside my body. I legitimately thought the championship game might be the last game I ever play of rugby. I, decided I was going to go out on my shield. I felt like individually, I played one of my best games. It was just that kind of emotional push, that knowing that I might not be out here again. Um, so this is a time, while these months are going by between the hospital and still going into April, that I would have liked to be an empowered patient, which is something we do here at CPATH. We empower everybody living with something to be their own strongest advocate, and it's obviously a huge part of what we're doing here at BMDR, which I quickly got connected with when I did receive my diagnosis, which we'll go through here in a second. So yeah, come May, I did feel sick again. I saw with my own eyes the discolored urine. So at this point, this is probably my first time where I was my own best advocate. I demanded a biopsy. There I am in the hospital. Yes, I am looking intentionally deranged, just the way that life works. One of my friends from that DC area texted me just to check in. Hey man, been a while. How's it going? I'm like, well, getting a biopsy right now, so been better. Um, I get the biopsy, I can see the results in the patient portal, but it's like speaking a different language for me, somebody in communications, not a scientist, not a researcher, not a doctor. So I'm calling my nephrologist every day. The assistant's starting to get frustrated hearing from me. She, and said, I already told you it's not a big deal. I'm like, okay, like you're telling me it's a minor case of CKD, that's not minor to me. Like it is, I am a word guy, I'm a communications guy. The words chronic kidney and disease put together to describe my own body are terrifying. And not really knowing, not having that in-person doctor's appointment to figure out what's going on was a very uncomfortable couple of weeks. You know, when I did figure out what, get in with a nephrologist, fortunately I was able, working at CPATH, to skip the line here, talk to Soren Fidelis, our executive director of our PKD consortium, as well as our CEO, Klaus Romero. If anybody knows Klaus's schedule, meant a lot that he took 30 minutes to talk to me about it. I got the diagnosis, the full rundown from my doctor, as well as the resources I had at CPATH. Right before Father's Day, that's when, on behalf of Father's Day and men's health awareness, I kind of wrote my first advocacy piece. So it's called When the Game Changed. My initial emotions as I was going through this were fear, confusion, anger. They're telling me it's minor, but again, chronic kidney disease does not feel good to describe your body. The anger, I think anybody that's gone through this, if you've been on the patient side, you realize that you've lost a little bit of control of telling your own narrative and sharing who you want to share with. Once news hits the Auntie News network, it's wildfire through the family. I get the unsolicited, how are you? Like, how's your health? I'm like, I didn't tell you. I didn't share this with you. Why are you asking me? Who told you? And you know what? How was the colonoscopy, Janice? Like, if you want to get personal about health information, let's get personal about health information. Um, so also following that, I mean, the real changes, the emotions are one thing. They're very real. I don't mean to dismiss those, but the physical changes, dietary changes, a lot of meat replacement with some other kind of protein supplement. So turkey sandwiches became PB&Js. You know, whole wheat bagels in the morning. One at most, animal protein based meal per day. Replacing dairy milk with almond milk. Just doing all the things that can take it easier on my kidneys to process my diet. Um, Med wise, I was quickly prescribed some high blood pressure medication, losartan potassium. And yeah, again, just kind of working and being inspired by CPATH, I wanted to quickly get involved, somehow be something of an advocate. That's when I got connected with the BmDR community. It was instantly a learning experience for me. I thought maybe people wouldn't hear from me that I wasn't, quote, sick enough, that since I had the minor IgA. I quickly realized it could not have been further from the truth with the the community workshops that we have here. Dr. Gary Friedman, who's on this call, quickly kind of took me under his wing so that we have regular check-ins every couple weeks. He can also, diagnose those charts that I have access to but can't read. Um, and he gave me almost a little bit of a glossary here of like how I could be empowered and enlightened person with living with CKD. I'm going to try to cut some time here, not read through this slide. Plus, I'm sure all the many people, the scientists, doctors on this call know these things better than I do. Yeah, what does this glossary mean though? What is Gary? What is Dr. Freeman trying to teach me? This is the big picture, right? My total glomeruli were 35. That's a good biopsy sample. 33 out of those 35 glomeruli are functioning as intended. This is very good, is what he's been telling me. And these are words that I like to hear. Very good. Um, Sorry, a little bit of delay getting to the next slide. Whoop. Oh man, sorry, I'm hitting a little bit of lag. There we go. All right, so the EGFR value of 122. This is good enough kidney function to last me 82 years. I will take that. I'm pretty sure, Gary, correct me if I'm wrong, that you guarantee me 82 years of absolute immortality. No, but I'll take the 82 years of good kidney function. And that's, of course, with the knowledge that, I have to continue to take care of myself, continue to prioritize my diet, realize that, walking around with 20 pounds of extra weight for rugby isn't the most important thing anymore. It's not about how much weight I can push with my chest. It's about what's going on inside my chest, keeping that blood pressure healthy. If I want to go for a run instead of a lift, I go for a run instead of a lift. Um, again, keeping those kidneys thinking like an organ, with everything I do, everything I eat, how I exercise, how I hydrate. That's a huge thing, especially if I'm going to continue to play sports. Going through 2 bottles of Pedialyte a week since diagnosis. Um, and Again, sorry, I'm hitting a little bit of a lag on there we go. So what's the point? Where do we want to get to? Where do we want to empower people living with chronic kidney disease? And this is, again, what's extremely important to both CPATH and the BMDR is empowering our people to be their best advocates, be educated. So what Gary and I would love to see are kind of at-home dipsticks, right? Almost feeling like that T1D model is T1D is kind of the poster child for an autoimmune disease where you can get a little bit of instant information. So you're not feeling right, you get this dipstick from CVS, no waiting on results. Get your updates in real time. Obviously, you still communicate with your doctor. You don't just take this and try to be your own doctor. But again, to tie this to the BMDR, the data collected by the BMDR represents the future of the empowered patient. I want to thank Gary. I want to thank the community. I want to thank you all for being on this call. I want to thank CPATH. Obviously, I didn't work. If I didn't work here, I wouldn't be speaking to you. But more importantly to that, just learning from people living with harder things than me and learning from them at CGIC, before that, the neuroscience annual meeting, people speaking so bravely, telling their stories about, young onset Parkinson's, young onset Alzheimer's. I would not be up here speaking freely without the courage that they gave me, hearing their stories. As soon as something happened to me, I wanted to follow their lead and just try to be a resource, be an advocate for everybody out there, whether they're going with CKD or just something else difficult, health-wise in their life. With that, I'm sorry I went over time. Katrina, thank you. I'll hand back to you and to our next speaker.
Thank you, Alex, for sharing your story. And we'll now welcome Dr. Joseph Bonventry, Renal Division Chief at Brigham and Women's Hospital and Professor of Medicine at Harvard. Joe, the floor is yours, and you should be able to advance.
Okay, great. Well, it's thank you for the introduction, and it's a great pleasure to be able to talk to you today. I'm as was mentioned, I'm the Division Chief Emeritus at the Brigham and Women's Hospital. More than 850 million people in the world have kidney disease, and most are not aware that they do. Injury to a kidney can occur due to many causes. Currently, clinical evaluation of kidney injury relies on measurements of the changes in serum creatinine or reductions in urine volume. These metrics represent the tip of the iceberg in detecting kidney injury, with the early stages of injury going undetected in the vast majority of patients. Acute injury can lead to chronic kidney disease, which can lead to accelerated cardiovascular disease and the need for dialysis or kidney transplantation. Furthermore, if someone has chronic kidney disease, they're predisposed to additional injury. So we are focusing on measuring molecules in the urine that enable us to tell individuals when they have kidney injury. What will the biomarkers of kidney injury allow patients and caregivers to do? Diagnose and detect kidney injury early in its course when interventions can be activated, and also will allow for the monitoring of injury over time to be sure that kidneys are not getting more inflamed or more damaged. These markers will allow us to predict which patients are more likely to develop acute kidney injury in response to a drug or a procedure. If a patient that's deemed at high risk, then special precautions can be taken to mitigate the risk. The dose of the drug could be reduced or avoided altogether. The drug can be avoided altogether. They will allow us to predict which patients with acute kidney injury are more likely to progress to chronic kidney disease or kidney failure, and that will motivate additional studies and also motivate clinicians to be more aggressive in monitoring the patient and potentially thinking about intervening with novel medications. Biomarkers can also serve as sensitive markers to confirm that a drug that is being taken to improve kidney function is in fact being effective. And this would allow for cessation of ineffective drugs and replacement with ones that may be more effective. Also, they'll allow us to predict which patients will have a positive therapeutic response to an individual drug or an intervention, so as to avoid potentially kidney toxic drugs in individuals who will likely not respond to them. The earlier we can diagnose acute kidney injury, the earlier we can take steps to minimize the chance that the disease will progress. The earlier we can evaluate our effectiveness of our interventions, then the earlier we can bring the right drug at the right dose to the right patient. The US government appreciates the importance of biomarkers as reflected in the 21st Century Cures Act. And I'll read it. Federally funded research should demonstrate direct and indirect clinical benefit to patients through innovative biomarker-based endpoints in clinical drug development trials and biomarker qualification for use in the clinical practice of medicine. As you have already heard, the Clinical Path Institute, CPATH, has been a driving force in progress in this area, resulting in the submission of a qualification application to the FDA, resulting in the first ever qualification of a kidney clinical safety biomarker panel by the FDA, specifically for use in early drug trials to assure patient safety. And more recently, as you've already heard, an application has been submitted by CPATH to the FDA to expand the context of use of biomarkers to allow the detection of response to drug-induced kidney injury in clinical trials, but to go beyond that and look at potentially efficacy and other aspects of biomarker possibilities in terms of interpretation. Now, in order to move biomarkers to routine clinical use, we have to provide additional evidence of their utility to the wider medical community. FDA qualification involves an iterative process whereby data is analyzed and additional data are also often required. And this is an iterative process, as I mentioned. Okay, so in order to obtain data, we have to have large studies where urinary markers are measured and correlated with patient outcomes. Fortunately, there are increasing numbers of studies where urine has been collected. The CPATH Data Repository, the biomarker repository, has been established to allow for the aggregation of the information from these studies so that the academic, pharmaceutical, and diagnostic communities can access and analyze the data and present their results to the broader community, including the FDA. But many of the very promising biomarkers have not been measured in these samples. Research and clinical colleagues and patient participants in these studies can be very persuasive in encouraging the measurement and analyses of these novel biomarkers. The more data that are made available, the more widespread the practice to measure them will become. With the encouragement of the FDA, it is more likely that new studies will collect urine and incorporate measurements of the most promising biomarkers included as key components of the protocols. Therefore, we ask our patients to be advocates in encouraging the study leaders to collect and analyze the data, but also make it available to the broader academic and pharma biotech community. Another key challenge is reimbursement. More data and qualification by the FDA will help to drive reimbursement for biomarker determination. So by systematically tackling these challenges, patients will benefit. More individuals will know they have kidney disease. Patients will be able to monitor their health if they have some kind of kidney problem. Fewer will have kidney complications of procedures or medications. Fewer, again, will suffer untoward effects of medications. Fewer will develop or progress to more advanced kidney disease and require dialysis and transplantation, and fewer will lose their transplanted kidneys to rejection. Thank you very much. It's been a pleasure to present this information to you.
Thank you, Joe, for your expert insights. And we will now turn to Andrew Palillo, Data Manager at CPATH, who will walk us through the available data sets. Andrew, over to you.
Thanks, Katrina. Our team has been joking lately that my role in this workshop is to tell you what meat we have in our meat locker. So as the Arby's commercial says, we have the meats. So on a more serious note, I wanted to take a few minutes to talk about the data sets that we have in the repository, data set details, data processes, and the BmDR workspace. So I won't take a whole lot of time to go through this slide, but this graphic shows the journey that an incoming data set goes on once it's ingested into the BmDR. On the left side, you'll see that we receive data in many different formats. Data are ingested through a secure platform into a quarantine space and inspected for PII, personally identifiable information, and then de-identified as necessary. That whole middle section illustrates that the data are standardized. We standardize the data in the BmDR to SDTM using similar variables and values as other BmDR datasets to make subsequent analyses as easy as possible. And then once we move out of that the big box in the middle, once approved, researchers can request access to the BmDR workspace on our data analytics platform, which we refer to as the DAP. All of our data in the repository can be pulled into the DAP for analyses. Researchers can also bring in their own data through an airlock into the workspace to interrogate BmDR data alongside of their own data. And all of this occurs to facilitate the end goal of creating actionable drug development solutions. Okay, so this slide shows what data sets the BmDR currently possesses. Our focus is on kidney biomarkers currently, but the BmDR's long-term goal is to accrue biomarker data from other organ systems as well. You can see this reflected in the data sets that we currently have in the repository. The top 8 are all kidney data sets, representing 746 subjects, and then the bottom 4 are other markers. Okay, so on this table, if you look down the left column, you'll see short descriptions of the data sets currently available on the BmDR. The mesothelioma data is not quite live yet, but it will be shortly. And then KPMP, you'll see, has an asterisk next to it. So KPMP is the Kidney Precision Medicine Project, and their data sets are not hosted on the BmDR, but are publicly available for download on their website. BmDR and KPMP, we are working collaboratively, and in 2026, our plan is to work closely together to find points of commonality and where our data complement and enrich each other. So this table shows which biomarkers are available in each data set and which assay platform was used to measure them. So the orange columns, those are the standard of care kidney biomarkers. The blue represent the biomarkers from the panel that Joe was referring to before. And then in addition to that, we do have some other non-standard of care kidney biomarkers in the platform as well. I know that was a lot of information, but before we move on, I'd just like to emphasize that by connecting the BmDR with the DAP, in other words, the repository with the analytics platform, we're providing researchers with access to a workspace where they can interrogate the BmDR data, bring in KPMP data, integrate their own data, and use open source tools that are integrated into the DAP like RStudio to independently pursue new hypotheses that can drive therapeutic advances. This is what the BmDR and the DAP offer from a technological standpoint, but now let's talk about impact. So our ultimate goal is to improve patient outcomes by being a resource to all the different communities within the greater nephrology community. So how does the BmDR impact those communities? What does the BmDR provide for the research community? Well, it provides quality kidney biomarker data in one place that's easy to interrogate for researchers. For the clinical and diagnostic communities, it increases understanding of biomarker performance and assay performance. For the pharmaceutical community, it paves the way for qualifications and publications that can inform early decision making. And earlier decisions can mean decreased costs. And for the patient community, it provides publicly accessible data for patients who have an interest in doing their own research. And the impact of the BmDR has already been seen in the nephrology community. Publications such as this one from Clinical Pharmacology and Therapeutics show how the previously mentioned biomarker panel has already had an impact on drug development. But the repository is only as strong as the data in it. So I'd like to close my time with a call to action. Any listener who has access to biomarker data, whether it's kidney or otherwise, is encouraged to reach out to myself or to Katrina to discuss the possibility of including your biomarker data in the repository. Thank you so much for your time and attention, and I'll send it back to Katrina.
Thank you, Andrew. It's now my pleasure to introduce Dr. Patrick Murray, professor at University College Dublin, who will present use cases showing how repository datasets can be applied. So over to you, Patrick.
Thank you, Katrina, for the introduction, and I appreciate the opportunity to be here and speak to you guys. I want to bring you greetings from Dublin, and I'm going to talk about the results of a later analysis of an earlier study of a European couple of cohorts looking at nephrotoxicity by what's called the Safety Consortium. The Safety Consortium was an industry academic collaboration funded by the EU that looked for validation and indeed discovery of new markers of kidney injury, liver injury, and vascular injury. I'm going to talk to you about two of the studies we went back and reanalyzed over 10 years after the original study in kidney injury from cisplatin and radiocontrast for radiology studies. Before I do that, I want to explain a conceptual model that I've been involved with for a number of years to help us think about how to look at biomarkers and look at kidney health versus kidney disease. It was developed originally by the Acute Dialysis or Diseased Quality Initiative Group, ADQI, for use in understanding AKI and how to apply biomarkers in that disorder. But it applies equally to drug-induced kidney injury. And it involves considering patients with normal kidney function, normal serum creatinine, normal GFR, and no evidence of kidney damage from markers in their urine as having renal health. The possibility of having worsening kidney function going down this y-axis with an increase in serum creatinine because of a reversible functional acute kidney injury where there's no damage to the tubules, but there's a need for hydration or some other intervention, maybe holding of a drug, for example, drugs that don't cause kidney damage but do cause decreased kidney blood flow. Versus another possibility, which is on the x-axis, that there is kidney damage, which in this quadrant is associated with the normal serum creatinine, a normal GFR, and it could be called at the moment subclinical kidney injury. Then finally, full-blown kidney injury with loss of kidney function down here, and development of evidence of tubular damage. This is often clinically called acute tubular necrosis, and is full-blown acute kidney injury, which can be drug-induced in many cases. And it's been useful to think about these quadrants and where patients belong within them, and thinking about how to manage their disease. So I think I'm going to go back to this as we go through the presentation. I'll point out one more thing, which is that if you look at large cohorts of patients coming into the hospital or coming into the ER, in every study that's been done, those that have normal kidney function and no kidney damage have the best outcomes. Those that have loss of both, with kidney damage and loss of GFR, have the most severe clinical complications. And those who have either one, a leak of damage markers or dysfunction without damage, have in-between outcomes. And this has been shown repeatedly. So I want to present a couple of studies. These were done over 10 years ago, but we reanalyzed a couple of the data sets because there was a bioresource which had the biomarker information and the clinical information. And I want to acknowledge Mike Strader, who's a clinical renal fellow who's finishing his PhD and worked on these projects, along with all the other authors of the first paper we published here, including investigators from Charité Hospital that collected a lot of the samples. The first example is using cisplatin, which is quite a toxic drug that's used for serious cancer chemotherapy. It was a cohort of 105 patients who were getting the first cycle of a high dose of cisplatin and had serial blood and urine samples, as you can see, compared to cancer controls who were patients who got non-cisplatin regimens or healthy controls. And we looked at all the same markers you've been hearing about. And the results were pretty conclusive for what the major questions were, which was, first of all, if you look at patients who've gotten cisplatin versus controls who have not received cisplatin, could you identify without knowing who was who, whether cisplatin had been given to the patient? And you can see that there are the kind of changes here between the two, looking at the peak biomarker concentrations percentage-wise compared to baseline, that all of these markers significantly differentiate between those who've received cisplatin and those who have not. The ones who have not are routinely lower, and the ones who receive cisplatin are routinely higher. So this was a very convincing sensitive detection of cisplatin exposure. So we already thought that we could probably detect subclinical AKI, and then certainly hoped that we could detect it before serum creatinine would, before the creatinine went up on top of the biomarker release. But what was the case? Well, in looking at the time course in the cisplatin exposed patients, you can see that it's encouraging that, first of all, serum cystatin C, which was mentioned earlier, which is an earlier and more sensitive functional marker, went up early after cisplatin exposure, much earlier than the peaks of BUN, a standard clinical test, and serum creatinine, probably the standard clinical test for kidney function, which didn't peak for a week. But you can also see that all of these other damage markers peaked far earlier, within a day or two, compared to the standard serum creatinine. And some of them in particular had very good sensitivity and specificity balances, such as kidney injury molecule 1 and osteopontin, for example, but a lot of them went up quite early. So this was very encouraging. So now we've got sensitive, but also time-sensitive detection of these markers, providing earlier detection of disease in the form of injury from cisplatin-induced kidney damage. So we probably already have evidence, even from a single study, that we can detect evolution from subclinical normal kidney function, because all these patients had normal kidney function going in, to subclinical AKI with damage that hasn't yet affected kidney function, through to full-blown acute kidney injury, which happened in about 20% of those who got cisplatin in this study. And primarily as a result of that study, both the EU, through the European Medicines Agency, and following that, the FDA gave support to the concept of using these markers in a panel, along with standard markers, things already available like albumin and total protein in the urine, as well as serum cystatin C, for the early detection of kidney injury in patients getting normal healthy volunteer studies, for the use of drugs, but also for consideration of use in more advanced phase 2 and beyond trials. Furthermore, as you've already heard, the FDA approved this panel, as you heard, which differs only slightly. Alpha GST isn't in this panel. There's another marker called NAG there, but a very similar approach to use of these drugs in early phase drug development. There's another study that wasn't prominent in the early letter of support, but we've also gone back and analyzed, and we haven't published this yet, and I have to ask people not to share the data on the slides where I show it for that reason. But we studied 121 patients with chronic kidney disease. So these are not normal kidney function at baseline, who were scheduled to receive a significant amount of radiocontrast for angiography, which is in chronic kidney disease patients, a common concern about superimposed acute kidney injury. Similar collections of blood and urine following the exposure, with controls including patients with CKD of similar severity who did not have angiography, or healthy volunteers. And the results are interesting, but different. So first of all, there was a statistically higher serum creatinine overall in those who got exposed to contrast versus not. And that's not surprising. About 17% of them got acute kidney injury by the clinical definition. The serum cystatins weren't significantly higher, which is a bit concerning, and there's a difference in sample size there. But what's interesting is that there's a different profile of biomarker release in this cohort compared to the cisplatin cohort. I mean, these patients start with chronic kidney disease, so different baseline. But in addition, the percent increases from baseline, which is what we're looking at here, were higher for instance, alpha GST, which was quite sensitive, and urine cystatin C, not as prominent for KIM1 or clusterin or osteopontin, but also quite positive for NGAL. And albumin and total protein were probably less impressive in this analysis than they were in the cisplatin analysis. But I want to point out a couple of other things that differentiate between the studies. First of all, I'll remind you that the increases in patients exposed to cisplatin were of the magnitude of several fold, 4, 6, 10-fold increases compared to baseline. Whereas the increases in what I just showed you in the contrast are no more than 100% increases. So they're lower grade increases. And there's always a difference between statistical and clinical significance. And I think there's probably something to learn from that. I also want to point out the time frame, which is quite different. But basically, the contrast-associated AKIs, which, as I said, was about 17%, the serum creatinine, by definition, went up within the first 2 days. So that's the peak. The controls were only sampled for 2 days because they didn't have any further creatinines after that. But what's interesting is, although the serum cystatin, as I said, didn't have a consistent pattern, the BUN also went up after 2 days. But now there's quite a difference in the profiles compared to what we saw with cisplatin. If anything, several of them are quite flat. There really isn't an increase of most of these markers, except perhaps NGAL and osteopontin. And these are small increases. These are absolute changes, and really quite a different pattern with less impressive injury documented. So in fact, what we're thinking about this is that this looks more like a functional syndrome than evidence of kidney damage. There isn't much evidence of tubular injury. There is evidence of dysfunction. It does reverse after the 2 days in these stage 1 cases. And in fact, there's a school of thought that, in fact, contrast-induced AKI may just be a decrease in renal blood flow due to renal vasoconstriction and a reversible phenomenon, and not associated with as much kidney damage as we thought. Obviously, that needs further work to explore. We've got lots of further work to do. We need to understand the course of these serial measurements. What documents resolution and progression, for example. Similarly, we want to know about longer-term outcomes, not just whether they get AKI at one stage or another of greater severity and recover within 7 days, but whether they go on to get acute kidney disease, which can last up to 90 days, or chronic kidney disease, which you heard about earlier. These are all important endpoints. But I hope you'll see that even just with a couple of small studies with well-characterized biomarker measurements and clinical outcomes, that we can start to learn a lot about drug-induced kidney injury. And hopefully with more collections and more statistical power, we'll draw firmer conclusions about some of the questions that we're raising here. Thank you very much for your attention. I look forward to the discussion. Thank you.
Thank you, Patrick. That was an excellent overview of the safety data. I'm gonna fly through some more data that you have here. And we'll move on to the panel discussion. But thank you to all of our speakers today. And now we'll move into the panel session moderated by Dr. Gary Friedman, one of BmDR's co-chairs. So please take it away, Gary.
Thank
And a
you,
reminder
Katrina,
to submit questions
and
through
thank you
Q&A.
all to all the speakers. I just want to reiterate that. Based upon some of what's been described here by Dr. Murray, what we're seeing is that on both sides of the Atlantic, 2 different consortia converged after looking at between 40, 50, 60, 100 different biomarkers. The 2 biomarker panels, as Pat Murray was alluding to, that were submitted to FDA and EMA for letters of support 10 years ago, the overlap there is almost 90%. Therefore, the data from that consortium, from the consortium on this side of the Atlantic, the Predictive Safety Testing Consortium, these now can be merged into the BmDR and be available to qualified researchers. As Joe Bonventuri indicated, the 21st Century Cures Act a decade ago really was the starting gun for this iterative set of processes that have led to reduced friction, meaning this process of going from biomarker discovery to biomarker qualification, instead of the 15 plus years that we've all been working on this, we want to help you to condense this down significantly. And therefore, there is at FDA and with EMA, there are biomarker qualification programs that are very, very clearly described on their websites. So as of now, we're just before the holiday season, and we wanted to describe some of the gifts that are waiting for you for those of you who take advantage of the data within the Biomarker Data Repository. First of all, as was mentioned by both the two of the speakers, the Critical Path Institute has taken two prospectively designed studies looking at the performance of the biomarkers, and those are being submitted to the FDA for review and qualification for use and understanding of acute kidney injury on an individual patient basis within clinical trials. Furthermore, as Andrew was alluding to in that beautiful diagram that I know is very intimidating, but please take a look at the BmDR diagram. This describes what is flowing in because of data contribution agreements with industry, academicians, and others. They are now contributing their standard of care biomarkers and not yet standard of care biomarker data, putting them jointly as de-identified data into the BmDR for interrogation. So instead of having one or two small studies, our anticipation is you will see quite a number of studies, probably closer to 10 completed studies within the next 6 months available for your independent evaluation. And we most highly prize your independent approach to this, and we'll provide the information through Katrina and others on how to be a qualified researcher to go through that very, very concise qualification process so you can look at that information. With all of this said, we now have two panelists that we are just delighted to introduce here. Dr. Frank Dieterle probably needs no introduction, but for those of you who may not be familiar with him, he is a global subject matter expert. He is at Novartis, and in terms of novel kidney safety testing, discovery, and innovation, Frank is on all of the major papers that you will see in bibliographies. Ashvina Dige is in leadership role at the Kidney Precision Medicine Project. Now, the Kidney Precision Medicine Project, for those of you who have not gone to their website, the KPMP Atlas is absolutely one of a kind. So patients who sacrificed to have a biopsy of their kidneys and then have serial blood and urine samples drawn over the years, you can see changes in biomarkers, some of which coincide with ours, others which have no direct connection to ours. But on top of it, it's a more robust DNA and RNA and further omics understanding of those patients' behavior longitudinally. Some of it based upon single cell omics from cells harvested from their urine. So with these two individuals on our stage for the next approximately 7 minutes, Frank wanted to pose something to you. If you could look at, the needs of regulators for governmental and non-governmental healthcare insurance payers, what are 2 or 3 actions that you see need to happen to make sure that these biomarkers, if qualified and when qualified, become available for the clinical management of individual patients?
Thanks, Gary. So Christmas is coming up, so it's 3 big wishes, and it's not only regulators, I make it more global. So first of all, the first wish is related to the content of the data repository. So currently we have a handful of studies in there. They have value, for example, establishing normal ranges in certain populations. However, to answer most questions and applications, you need a lot more data and a lot more variety of data. So my first wish is that we can increase the amount of data in there, and it's important for pharma to understand the value, because if you want to contribute, first it's an effort, it's an investment. You don't have a direct return of the investment. And there are also often concerns by pharma. Can we share data? I think most of the concerns can be alleviated. Data can be shared anonymized. You can decide what to share. Let it be only a comparator arm. Let it be only terminated projects. So my first, and I always compare this data repository with open source development. So first it starts very small, limited contributions, which are critical. And once you reach an amount of data or in software amount of features, everybody recognizes the value, and suddenly contributions come in by themselves. So the first wish, a lot more data. The second one is availability of tools. So if you want to implement these tests in routine clinical practice, you need tools. And I talk here about in vitro diagnostic tests. So far, the diagnostic companies have not engaged in these kidney safety biomarkers, and there are several reasons for it. The first one is kidney safety is a pretty undefined wide disease area. If you develop an oncology test for a certain mutation, you have a very clear indication, very clear population. Whereas, for example, drug-induced kidney injury is the entire universe of nephrotoxic drugs, which no company can tackle themselves. So they need to join these collaborative efforts, which currently are being driven by pharma and academia. So they need to be part of the big game. And also diagnostic companies, they have the challenge of economics. So these tests are currently not being reimbursed. And that brings me to my third wish, the recognition of the value of these biomarkers. So if you want to implement these tests in clinical routine practice, you need a decent reimbursement. Currently, standard of care is serum creatinine, it's few cents, and payers need to have the evidence that there's a health economics value of suddenly reimbursing a lot more expensive tests. And that's where a lot of players need to work together, because it's a real value for patients and for health economics. It's not preventing AKI, it's preventing long-term chronic kidney disease or reducing the risk of chronic kidney disease. And that's where you need to contribute not only AKI studies, you need to combine chronic kidney disease studies, epidemiology studies, real-world evidence, all in one place. So a lot more for the biomarker data repository. And that's my third wish, a big collaboration, and that everybody contributes for the value creation and therefore enabling also reimbursement by payers. So that's my third wish. Back to you, Gary.
Frank, thank you so much for those. And what we can say with confidence is because of the hard work of the people you see on the screen from CPATH, the number of studies that when you explore the biomarker data repository online, you will also see the data analytics platform. So you will be able to look at the growth of the number of patients from just a few hundred to more than 1,000 over the coming months. But you'll be able to see that for yourself. And once you get qualified, you'll be able to access that data so that you can analyze independently and publish independently. As far as the availability of those data, in terms of companies that could be looking to develop in vitro diagnostics, there are avenues for which that can occur. So, Frank, we will be giving more information based upon your holiday wishes. You'll see those in publications that come out monthly from the Biomarker Data Repository and CPATH. Thank you so much. And now, looking into the future, Ashvina Dige is here. Ashvina, are you still on the line?
I'm here, Gary.
Okay, thank you so much for being here. You know, Ashvina, for many of us, who have been watching KPMP just crank out incredible information, and now the Atlas. We're very excited because KPMP, one of the things we see is not only understanding the dynamics of what is going on at the time of injury, acute or chronic, but it also offers this single cell omics capability that has a possibility of prognosticating. Right now, we have very few capabilities that allow us to prognosticate accurately in terms of functional recovery versus scarring. So, with all of that said, knowing the importance of the KPMP mission, can you let us know 2 or 3 holiday wishes for what you would like to see in 2026 for KPMP to continue its mission being accomplished?
Absolutely. So we are 8.5 years into our project. We're funded in 5-year cycles. So my first wish would be to make sure we have another 5 years to accomplish what we're doing and really get data out there that companies can use to recognize real patient outcomes in the future. I think we've made a lot of progress and I'm very proud of what we've done, but we need more time. That would be my first wish. My second wish is funding for more biomarkers. We're funded internally to establish a core set of biomarkers, but we need help in doing the other proteomic biomarkers that we've funded. We have biomarkers out there and whole genome sequencing data for our first 330 participants, but we are nearly at 700 participants enrolled now. So we have data biomarkers potential that we're sitting on. And we really need partnership, I think, with industry to realize that common goal and get the additional data out there. And the last thing I would say is to just better socialize KPMP outside of their regular channels. And we're very grateful to our collaboration with CPATH, who is helping us get the word out there about KPMP data. We have over 20,000 data sets available right now. And you can see on our statistics just views popping up and ramping up from countries all over the world. And I think it's really important to continue for people to continue to use our data and also apply to get our biomarkers for novel biomarker use.
Thank you so much, Ashvina. Frank, Ashvina, thank you so much for sharing your wishes, and we hope to help to reinforce that your wishes need to be granted throughout the coming weeks and months. So thank you. Katrina, back to you.
Thank you, Gary, and thank you everyone for staying on a couple minutes later than we had planned, but we had a lot to get through and we reviewed a lot of material today. Thank you for all the attendees for joining us. Thank you for the panel and all of our speakers. Again, this was the first in a series of workshops, so we have two new workshops planned for the next year, and we have lots of ideas, but we want to hear from the community and from you as to what's most important to you to hear about. So there will be a survey that pops up after this webinar that you can submit questions or topics to us through. And additionally, if you have any data to contribute to this effort to advance kidney biomarkers, please contact us and we'd love to work with you. But again, thank you everyone for joining us. This recording will be shared and posted to cpath.org afterwards. And we hope everybody has a great holiday and we'll see you in 2026. Thank you all.